Okay. Good afternoon, everyone. Welcome to Agri-Food Conversations, brought to you by iSelect Fund, the Van Trump Report, the Yield Lab Institute, and Family Farms Group. My name is David Yoakum. I'm an associate on the iSelect Fund Ventures team, and I'm excited to welcome you all to the discussion today. Agri-Food Conversations is all about driving innovation in agriculture. Each month, we highlight a specific theme, including emerging topics such as soil health, plant genetics, vertical farming, and aquaculture, to name a few. This month's theme is inputs and biologics, and on today's call, we're joined by Anna Rath, President and CEO of Vestaron. Vestaron is embracing the power of peptides, starting with a class of compounds that kills insect pests efficiently, but is safe for humans, birds, fish, pollinators, and the environment. As part of this, the company has developed a proprietary platform for peptide optimization and fermentation-based peptide production that will allow development of a wide variety of biologic crop protection solutions. Now, each of you knows that customers are more likely to succeed with the right network of customers, talent, investors, and advisors. We've invited you to this call because you're some of the smartest, most talented people in Vestaron's market. You are potential customers for Vestaron's products and services. You have built a company similar to Vestaron, or you are a sophisticated business person or agricultural professional who understands the market and the challenges and opportunities Vestaron may face. Before we get started, we have a quick poll question to get a better idea of who we have on the call today. Please take a few seconds to answer. And while that poll is running, a few process comments. We are not soliciting investment. This presentation is to provide information to help Vestaron find customers, mentors, and other strategic relationships that can help them grow their business. You are all on mute. You can use the chat window to ask a question at any time. This presentation is being recorded and will be available for replay. And so with that, and without further delay, I am pleased to introduce Anna Rath, President and CEO of Vestaron. Anna, please feel free to take it away.
Great, thank you very much. So first, here's this is the official disclaimer, right? So I'll leave that up for just a second for everybody to take it all in. Undoubtedly, you'll very speedily read all that fine print. And here is the contact information for iSelect and Van Trump and Yield, should anybody have need to reach out to them. So on to Vestaron. So exactly as David said, what we are doing is bringing biologic crop protection products to market that have the efficacy of synthetics, and that's a fairly novel thing in agriculture. So let me dive into why we think that is a really big deal and what we're doing about it. So our view is that crop protection is an industry that is primed for disruption. It's a large market, as I'm sure many of you are well familiar. We think that it looks very much today the way pharma did in the 1980s, where the history is predominantly one of small molecule development being done in-house by a handful of majors. But the same wall, right, that pharma hit back in the 1980s with that model appears to be being hit now by the ag chems industry. In particular, the good news about these small molecules is that they do what they're supposed to do. They bind to their target receptor and they have a reliable interaction when they do that. The problem is they tend to be a little bit promiscuous. So they often also bind to things that they're not supposed to target. And in the human health world, that can lead to unintended health consequences. In the ag world, that can lead to environmental harm, harm to beneficial insects, harm to humans and other mammals, harm to fish, harm to birds, etc. And so what happens when we realize that our small molecules, at least some of them maybe have some issues? So the first thing that you get is blockbuster products being pulled from the market. The next thing you get is increasing regulatory scrutiny and longer, and as a result, longer product development timelines and greater product development costs. And then finally, as a result, you get a declining pace of innovation. You can't have successful crop protection without, or you can't have successful agriculture without crop protection, right? So genetics is an important part of it, but so is crop protection. So here you see what the yields would be of the major row crops in red if there were no crop protection. The orange is the addition to yield, the protection of yield that comes from the crop protection, and then the blue is the expected sort of remaining loss, right? Like the room we still have to grow to get to perfection, if there is such a thing. So here, the chart on the left shows you the trajectory of novel actives in crop protection. And again, you can see, the industry was on a great trajectory up until around the sort of late 90s, when you did start seeing this increasing regulatory scrutiny, and that led to a significant decline in the pace of novel actives being introduced, and along with that, significant increases in the cost of developing a novel active. The EU is sort of in the lead on all of this, on banning harmful pesticides. So what you see on the left is what existed as solutions for farmers in the EU back in 1991. So all of those, right? All of those were available to growers as of 1991. As of today, everything in blue there has now been banned from the market. So you can see that growers today only have about 40% of the solutions that they had in 1990. And then the bar on the right says, okay, well, how has the ag chems industry done on creating additional solutions to sort of fill that gap? And what you can see is that if you take the red bar on the right, the new solutions that have been approved since 1991, and add it to the red bar on the left, you're still only at about 60% of the total solutions that growers had to them available as of 1990. And that is a particularly tricky thing as it relates to pesticides, because of course, if you keep trying to kill a pest with the same thing over and over again, eventually that pest will develop resistance. So you can't have this narrowing set of available solutions. So we really believe that as a result of all of this, a shift is needed in crop protection. So chemical pesticides are great at doing what they're supposed to do, but as we talked about, they can have these negative consequences. And it is increasingly difficult to commercialize new chemical actives for all the reasons we just discussed. And because of the declining number of existing solutions, those existing solutions are facing resistance challenges. So what are we doing? What we are doing is using peptides. Peptides are small proteins. And we believe, leading a peptide-based revolution. We really do think that it is revolutionary to ag chems, providing growers with novel, effective chemistries that address proven targets, but overcome existing resistance issues, and do that while offering a very favorable safety profile for workers and for beneficials and for the environment. So that is what we are attempting to do. And we are the first company bringing peptide-based biologic products to crop protection. And it is not futuristic, it's not theoretical. Our first one is on the market. So why did we choose peptides? So in the pharmaceutical industry, when you say the word biologics, you mean proteins, right? And again, peptides are just small proteins. Why did pharma migrate from small molecules to proteins specifically? They did that for two reasons. The first one is that proteins can have the same level of dependable interaction with your target, with your receptor, that a small molecule does. But unlike small molecules, proteins are bigger, and so they have bigger binding sites. And so they're more specific in what they will and won't bind to. And so they are more likely to successfully target your desired receptor without also targeting things that they're not supposed to bind to and creating negative side effects as a result. The other reason the pharmaceutical industry shifted to proteins is that when they are being metabolized in the body, or when they're being metabolized in the soil and the environment, proteins degrade to amino acids, right? So they're basically a nitrogen source. You have absolutely no concerns about any of the kinds of secondary metabolites that either a human liver or anything in the environment might turn any of these small molecules into. And so from an agricultural perspective, why peptides? Because they're safe, right? Because they are basically just amino acids at the end of the day. So they're safe for humans and mammals, safe for fish and birds, safe for beneficial insects. We have a 0-day pre-harvest interval. So if you have a thrip outbreak today, you can spray your crop today and put it on the grocery store shelf tomorrow. They are sustainable because it's a fermentation-based manufacturing process, so entirely renewable. And as I already mentioned, degrade to amino acids in the environment, so very environmentally friendly. And perhaps most importantly, from a sustainability perspective, is you have to enable ag to remain productive. And for that, you need new solutions, ones that meet the increasingly high regulatory bar. And so we believe that our solutions are enabling for sustainable agriculture. And that's a big part in our mind of what sustainability means in this context. As we've talked about, and I'll show you some data later, same efficacy as small molecule synthetics, right? Same direct, known, reliable chemical interaction between the peptide and its target receptor. And then interestingly, and we're in the midst of some academic collaborations to help prove this point out, but there is literature in other areas that suggests that when you have these larger molecules with these larger, more flexible binding sites, it actually might be more difficult for resistance to develop. And that's because a single point mutation in the receptor of the insect might not interrupt binding with these larger peptides in the same way that it could with small molecules. So stay tuned for some more insight on that front, but we think that could also be a really interesting underpinning to why peptides are really a very compelling alternative for pesticides. So unlike in pharma, where when they say biologics, they mean proteins, in ag, when we say biologics, we mean a lot of different things, right? And things called biologics have existed in ag for something like 30 years. But it's important to understand that not all biologics are equal, right? That they are not all the same thing. So the vast majority of biologics in ag to date are either whole microbe products or even, sort of multi-microbe products, or they are plant extracts. And the issues with both of those can be that they don't have a single knowable, reliable mode of action the way you get with a, small molecule in a receptor or peptide in a receptor. You don't know exactly what's happening. You just know that these things kind of work, and maybe you can suss out some of the ways that they work, but it's not the same kind of drug to target interaction that you get with a small molecule. And as a result, often they have lower efficacy relative to synthetics. You just don't get the same level of pest control. And then finally, also, you can often have issues with their ability to be drop-in for existing agricultural practices, right? So do they have the field stability? Do they have the pH stability to go into a tank mix, et cetera, et cetera? The consequence of all of this is that historically, biologics have rarely ever exceeded 10 million in annual sales, whereas your average synthetic insecticide has 75 million in annual sales, and the leading synthetics can have $1 billion in annual sales. And so what we think we are doing here is bringing forward to the market molecules that have that synthetic level potential, right? And thus similar revenue potential, but with the development costs of a biologic that are more like 10 to 20 million in aggregate versus the 300 million required for a synthetic. And obviously, those are compelling economics. So before Vestaron, as I've sort of gone over, there were really two classes, right? There were the synthetics and there were the microbials. And the good news about the synthetics is they did what they were supposed to do. Bad news was time to market and cost to develop and sometimes safety profile, right? On the microbials, really good on cost to develop, time to market, safety profile, but didn't always get full control, don't always have stability in the field, and production can be variable. And so we really think that in many ways our peptides are sort of the Goldilocks, if you will, of pesticides. That we have sort of the favorable characteristics of the biologics in terms of cost to develop, time to market, safety profile, but we have the favorable attributes of the synthetics in terms of production, costs, and product stability. So let's talk about our first peptide. Oh, and apologies, I've got some barking dogs in the background. So Spear is our first peptide. It was launched in earnest this year. And interestingly, it was the first novel nerve and muscular mode of action in all of insecticides in more than a decade. So for those of you not familiar, there is a committee called the Insect Resistance Action Committee And it is the job of this committee to decide if your insecticide works the same way as an existing chemistry or works in a novel way. And that's important because insects do develop resistance to existing chemistries. And so if your chemistry is just a me too, right, has exactly the same binding site on the same receptor as some existing chemistry, then insects that are resistant to that existing chemistry will also be resistant to your chemistry. And so an amazing thing to me is that in the whole history of insecticidal chemistry, there are only 32 modes of action. And of those 32, 14 are nerve and muscular. And those 14 turn out to be far and away the most important. So those 14 nerve and muscular modes of action account for 80% of the global $18 billion insecticidal chemistry market. But there hasn't been a new one since 2007 in the whole rest of the egg cant industry until we launched Spear in 2018. So it's a really big deal for the industry that we have brought this product to market, and that not only is it a novel nerve and muscular mode of action, but again, it is that with all of the safety and environmental profile of a biologic. So this first peptide, Spear, targets the nicotinic acetylcholine receptor, which is the same receptor that is targeted by the neonics and the spinosins. And so together, that receptor is responsible for about 30% of the global insecticidal chemistry market. But unlike, we do bind to a different site on that receptor, so that's why it is a novel nerve and muscular mode of action. And unlike the neonics, many of which are now banned, Spear is not harmful to honeybees or any other beneficials. And we have no cross resistance with either the neonics or the spinosins because of that novel binding site. So when I told you there were 14 nerve and muscular modes of action, those are actually across 8 different receptors in an insect, 6 of which are most important. So it turns out 6 receptors in an insect account for, oh, I gave it to you wrong. The 14 novel nerve and muscular modes of action account for 85% of the global insecticidal chemistry market. The 6 receptors that are most important, that are targeted by those nerve and muscular modes of action, those 6 receptors account for 80% of the global insecticidal chemistry market. And we believe that we can come up with 1 or more peptides that targets each and every one of those, sort of key multi-billion-dollar drug targets in effect. So let's talk about Spear. Spear is in the class of ICs, right? ICKs, inhibitory cysteine knot peptides. It's about 40 amino acids long, very broad pest spectrum. We've already talked about the pre-harvest interval, also a 4-hour reentry interval, so very safe, no regulated residues, and no phytotox. We have brought it to market in 2 initial forms. One is Spear Lep. This is an ingestion kill, so it's a foliar spray and the bug eats it and it kills the bug that way. That's targeting lepidopterans in high-value field crops. And the other is a contact kill. So this is aimed more at your soft-bodied insects, right? Your thrips and whiteflies and aphids and mites. And it is a miticide as well as an insecticide, which is an unusual thing as well. So just to show you some data to sort of prove out the point that, in fact, we do control every bit as well as the synthetics. So here you're seeing puddling moth and oriental fruit moth in apples. And so your left-hand side is your untreated control. And then you have the Spear treatment alone. Then you have Spear alternating with chlorantraniliprole. And then you have a more traditional synthetic rotation of acetamiprid and chlorantraniliprole. And what you can see is there is obviously no difference between the ability of each of those treatments to control the insects. Similar, oblique banded leaf roller. So here you've got your untreated control. You've got your Spear Lep at a higher rate. You've got your Spear Lep at a lower rate, alternating with spinetoram. And then you've got spinetoram again by itself. And again, no significant difference in the ability of the different molecules to control the insects. This data was actually surprising even to us this year. So this is Naval orange worm in almonds. And So this is spear, our spear treatment versus a conventional, and the conventional, we did both versus Besiege and versus Intrepid, and then we rolled that data together to produce this chart. But there were multiple time points taken here. So the way this trial worked is you sprayed in the field, and then at various intervals after that spray, the nuts were harvested and taken into the lab and put in buckets with navel orangeworm larvae. And then you saw the level of control that you still had at those various time points. And so it was, 5 days, 14 days, 18 days, 20, etc. Right? Lots of time points on this. But the point that I want you to take away here is that even out to 30 days after treatment, spear had efficacy on par with Besiege and Intrepid. What other biologic can do that, right? I mean, that's really a novel thing in the world of biologic pest control, and so something that we and the almond growers are all pretty excited about. Here's another example of a situation in terms of what starts happening when you start seeing some resistance, right? So here you're in a field that had some resistance to radiant, and so you can see the spear at a low rate, right, at 1 pint per acre. It offered some control, but not full. At 2 pints per acre, pretty good control, and you can see that relative to radiant, which really didn't offer very much control relative to the untreated control because of the resistance that you're seeing in that field. So just at a high level, this is who we are. We're about 40 people. We are, I guess, wow, now 15 years old. I've only been with the company for 2.5 of that, but it had a great history building, obviously, a very robust technology platform before I joined to really help it become a much more commercially oriented company. Our headquarters are now in the Raleigh-Durham area of North Carolina, but we've maintained a research site in Kalamazoo, Michigan, which is where the company was originally based. And our investors are more or less a who's who of kind of the ag tech investing world. Probably most of those names familiar to you. And then in case you don't know them because they're not ag tech, Novo Holdings, which is the parent company of both Novozymes and Novo Nordisk. And so they are a large life sciences investor, and we're interested in kind of taking their first step into a slightly adjacent space, and found us interesting as an opportunity to do that. And so just in summary, so as of today, we are very much focused on these time-rich peptides for insect control and our ability to re-drug, right, all of these critical synthetic insecticide target receptors with this safer, more environmentally friendly class of molecules. And I will tell you that we expect our second molecule to come to market at the end of 2022, our third in 2023, and our fourth in 2024. So the pipeline is very clear at this point. And then in the future, we think that based on our what we know how to do in terms of optimizing these peptides and manufacturing them, that we can sort of spread from insecticides into other areas of crop protection, right? Fungicides, antibacterials, etc. And so that's sort of piece number one. Piece number two is there are some other interesting applications out there of insecticidal peptides, right? Consumer garden, animal health, etc. And then finally, this peptide production system would not be limited to crop protection, but could actually be used to produce peptides for other fields, pharmaceuticals, etc., as well. So that is where we are and where we see ourselves going. And so with that, I would happily take any questions.
Wonderful. Thanks, Anna. Really excellent to hear all the progress, and obviously a kind of a groundbreaking, particularly on the regulatory side, product. So really appreciate you sharing everything with us. For anybody in the audience, if you do have questions for Anna, now's a great time to ask them. The best way to do that is by submitting a question through the Q&A box as opposed to the chat window. That way we can file all the questions in the order that they are received. So please feel free to ask questions, and I will answer them as they roll in. I am curious, Anna, just to kick things off, in terms of this being the first peptide being used in ag for crop protection or pest control. What does that mean for others out there who want to develop peptides as well? Is it a rising tide raises all boats? Are there still challenges for those companies that are trying to develop those products? And just tying into that, one thing that I noticed is that there are so many desirable properties about this specific peptide that it almost makes it hard to believe that there are so many others out there that would work so well. And so can you speak to those 2 points about others who might benefit from now there being a precedent, and then whether or not there are many other modes of action that can be pursued?
So we don't see a theoretical limit to the modes of action that could be pursued. We think that this is every bit as robust a class of molecules as is small molecule synthetics, right? So we don't think there's a limitation to the class. I would say for anybody wanting to sort of follow in our footsteps, the pithy way that I will say it is that they would have to make all the advancements that we did over the course of 12 years in terms of learning how to manufacture these peptides, learning how to optimize the architecture to make them stable, viable commercial products, and then figuring out how to get them into and through insects successfully. And then after they figured out how to do all of that, they'd have to figure out how to navigate around our patents.
Well, that sounds like a lot of work to have to do. We've got a couple of questions here from the audience. The first one is, what is your current time to market cycle, example from concept to commercial launch?
Yeah, so I mean, at this point, N is 1, right? We've commercialized 1, so it's not a perfect view, but I think best guess would be probably 4 to 5 years from conception to initial commercial launch in the US, right, where we have an 18-month regulatory process, right? Much longer, for instance, in the EU, where first you have to wait for your regulatory slot, right, and then the process itself is probably going to be somewhere between 2.5 and 3.5 years, depending on how good a job you do in the package.
Yeah. The next question we have: do you have proprietary fermentation technology in facilities, or do you use CMOs for fermentation?
We have proprietary fermentation technology, but we do use CMOs to implement that technology. So much of what we have right now is protected largely by trade secret, but we do expect to be filing some patents on some aspects of the manufacturing process in the near future.
Got it. Do you have a sense of how you'll scale production for commercial products?
I mean, we're already in 100,000 liter fermenters, so we're pretty much scaled. Now, really what we're doing is, more and more batches, as well as bringing on, multiple manufacturers to make sure that our risk is hedged should we run into any issues with any particular one.
Got it. And then another question we have here: what is the cost of producing the peptide?
What I can tell you is that at price parity with leading synthetics in terms of our price to the market, and then with distributor margins and everything taken out, our cost of goods, or our gross margins, let's say, are right around 40% today, and I see ready opportunity to get those into probably the low 50s just with process improvements and volume increases, and then into the low 60s with some additional improvements to strain and formulation to reduce usage rates in the field.
Got it. I'm going to pause here to see if there's any further questions from the audience before we move to the next part here. The question is, another question that just came in is, how do you source your peptides? Or perhaps answer differently.
Yeah, no, I can take that. So it turns out that the class of peptides that we work on is one that has been known to academics and industry for decades. People have always known that there were these insecticidal cysteine-rich peptides out there. But the problem was nobody could figure out how to manufacture them. Nobody could figure out how to make them stable in the field. Nobody could figure out how to get them through an insect. And so what Vestaron did in its first sort of 12 years of life was overcome some of those things that nobody else had been able to do, and then amass some IP around that. There is still a good bit of low-hanging fruit that we can sort of pluck now that we kind of have our secret sauce figured out, right? And use that secret sauce to turn those natively existing peptides into things that can be viable commercial products. But then, even once we exhaust that, then you just turn to, screening of libraries and de novo synthesis and so on. So there's no sort of inherent limit to the source material for all of this.
There are two questions here that sort of relate to traction. So I'm going to give you an opportunity to sort of answer them in whatever way you feel like you can, in terms of, what traction looked like in 2020. And the other question being, how many acres have you treated since the introduction of Spear in 2018?
Yeah, so Spear was approved in 2018, but I would say it wasn't really introduced until 2020. So I would say that both of those questions should properly look at 2020. And then what I will tell you is I haven't found anybody who doesn't believe that what we did this year was the highest level of revenues that any small biologics company has ever achieved with the introduction of its first product ever. And we did that with the headwinds of COVID. So you can guess what that might mean in terms of numbers.
Good answer. Excellent. Well, Anna, the last question that we have for you is, what can the audience do to help you out here, and how can they find you?
Yeah, so obviously, if any of you are in the space of specialty crops, right, fruits, vegetables, tree nuts, etc., we'd love to talk to you about Spear. We have a pretty good distribution network set up, but if any of you are distributors, certainly also welcome to, interested in having those conversations. We are not fundraising right now, but I'm sure we will be again, because that's how it works. So if anybody wants to have that conversation, happy to do that. And yeah, I think those are sort of those would be the major points.
Excellent. Well, Anna, thank you so much for joining us today, and congrats on all the progress to date. I'd also like to thank the audience for your active participation today and your great questions. To let everybody know, we host these calls every Thursday at 3 p.m. Central Time. And you can register for AgriFood Conversations by going to agrifoodconversations.com. And if you know someone who would like to watch this webinar, feel free to share it with them directly. A replay will be available in the next 24 hours and shared with you via email. And you can also direct somebody to agrifoodconversations.com where they can view as well. But otherwise, Anna, thank you so much. Thank you to our audience, and we look forward to seeing you next week.
Thank you. Appreciate the opportunity.
Mm-hmm.